Hi, my name is Lauren Shechtman. I'm a third year grad student in the Developmental and Stem Cell Biology program at UCSF. I'm originally from Chicago, and I did my undergraduate studies at the University of Illinois at Urbana-Champaign. And it's here that I found my passion for research working in Dr. Bruce Fowkes' lab. I then took some time off working as a lab tech at the University of Colorado Anschutz Medical Campus in Dr. Linda Barlow's lab, which seeks to understand the mechanisms behind taste receptor cell development and regeneration. And it was during this experience that I really became passionate about stem cell biology and where I saw the power and potential that this research may have regarding potential personalized medicine treatments. So now as a graduate student in Dr. Jeroen Roos's lab, I study the signals that govern cell fate decisions and the regenerative response in the small intestine. All right, so the epithelium of the small intestine is where I focus my work on, contains differentiated absorptive and secretive cells that are located in these epithelial structures called villi. But in order to maintain proper function, these cells only live about 3 to 5 days and are continuously replenished by intestinal stem cells. So stem cells actually listen to these signals, these epithelial and developmental signals, which tell them to become any of these differentiated cell types in the gut. But my work focused on the rare chemosensory cell, the tough cell, a cell that's responsible for initiating type 2 immune responses. So for instance, tough cells actually sense worms in the gut and can mount an immune response that simultaneously clears worms and increases tough cell number in the gut. However, it's still unclear whether or not any epithelial or developmental signals that I mentioned, like you saw earlier, are capable of driving tough cell fate. We found that when we knock out this gene called RAS-GRIP1 in mice, that their guts have fewer tough cells than control mice that have functioning RAS-GRIP1. And so you can see here, tough cells are these little green cells here, and there's much fewer in knockout mice relative to controls. And then importantly, mice deficient for RAS-GRIP1 are actually unable to clear worm infections as quickly as control mice. Conversely, when we overexpress RAS-GRIP1 in mice, this actually enhances tough cell production, as you can see in this image here. Additionally, these mice are capable of clearing worms much quicker than wild-type controls. So here in these overexpression mice, they don't have as many worms as the wild-type control. We've transcriptionally profiled tough cells from control and knockout mice, and this has resulted in clues that have guided us in understanding the signals that drive tough cell production. So we're currently using organoid technology and various commercial drugs to test these findings. Finally, I would overall like to thank the ARCS Foundation for awarding me this fellowship and providing me with the opportunity to share my work.